Thank you very much for inviting me to give this talk. So I'm not going to show any pictures of Diamond, so I thought there was no better praise of Diamond than just showing the work that was generated through the use of the Diamond facilities. And so what I'm going to tell you about is how those hair that you're seeing here, bacterial hair, are produced. So this is a bacterium that you're seeing here, and you can see those appendages here, hair-like fibers, which we call pili. And you might wonder, why should we study those things? Seems pretty useless, but it turned out that those pili are important. What I'm showing here on the left is the detail of a bacterium. So this is what I'm showing here with my pointer. A bacterium, it's a uropathogenic Escherichia coli, so it's a bacterium that infects the urinary tract in humans. And the thing that's starting to surround the bacterium is the bladder epithelium. So what this bacterium is about to do is to get into a bladder cell. And obviously, this is what we call cystitis, and you might know or might not know cystitis is a very common problem, particularly in aging or aged people. And so we're trying to understand how this bacterium locates to the bladder and starts infecting the bladder. And really crucial to this process are those things that you're seeing here. So those are the, I'm trying not to tremble too much here, but I'm not succeeding terribly well. I have to use two hands. So what you're seeing here are those little bridges that you see are the pili. And if we knock out the genes that produce or participate in the production of those pili, the bacterium is totally harmless. It's unable to recognize the bladder, so will not attach to it, will not recognize it, and will not cause infection. So those pili, in that sense, are really important. They're the ones that are going to recognize the bladder, allow the bacterium to locate to the bladder, and obviously a number of processes will result from this attachment. And those processes are happen in the bladder cell. And I don't want to go into the details of those processes, but they lead to this invagination that you're seeing here. So basically, the attachment of this bacterium through the pili to the bladder cells trigger in the bladder cell a reaction that will lead to the engulfment of the bacterium. And obviously, that's what the bacterium wants. It wants to be intracellular. It wants to feed on the cell. So this is really how the infection works. So we've been preoccupied for many years trying to understand how those pili are formed. And so there are a number of assembly pathways for those pili. There are polymers of protein subunits, so of proteins. And although in nature there are 3 different assembly pathways for those pili, we study one of them, which we call the chaperone pathway. So I'm going to tell you about the chaperone pathway. Those are pictures of the pili, of the 2 types of pili. One we call the P. pilus, and any bacteria that will produce this kind of pili, the P. pili, would want to be targeted to the kidney. And so that causes pyelonephritis, or infection of the kidney, and that's a prevalent disease in newborns. And then, as I told you just now, just before, the bacteria that produce those type of pili, the type 1 pili, are targeted to the bladder, and they cause cystitis. And again, between the 2 of them, they are responsible for 80% of urinary tract infections. So I'm going to try to explain in 5 minutes how they are produced, at least the state of knowledge before we got into the field. And so what I'm showing here is a schematic representation of a pilus here. As you can see, it is anchored to a membrane, and this is called the outer membrane, because where those pili operate in a particular class of bacteria that have two membranes. So many bacteria have two membranes. They have an inner membrane that you're seeing here, and then they have an outer membrane. And you can see that the pili originate from the outer membrane. So the pili are polymers of subunits, protein subunits, as I told you, thousands of them, but there are only 6 types. So that means that, at least for the type of pili that I'm showing here, 6 types of subunits, and I call them G, F, K, A, and H. And of course, each of those subunits are present in different stoichiometry, and this why you end up with thousands of them. But let me explain a bit more. You have this G subunit here, which is always at the end. And this is the business end of the pillars. It's the one that will cause the attachment, the binding, and the recognition of the host. You have this, so this is only one copy of this G subunit. You have this F subunit, which is also one copy. And then what you find is usually a very small polymer of 5 to 10 subunits of this yellow thing here, which is called E. Now another subunit, one copy of this one, the green one, and really the bulk of the pillars is formed by this subunit in cyan, for which you have 8,000. Okay? And then at the end, on the other side of the outer membrane, on the other side of this membrane, you have this subunit H, which we call the termination subunit. It's always plugging the whole thing here. So always the last one to be incorporated in the pillars. Now let me tell you how this works. All those are obviously, I told you, they are proteins, so they are obviously produced on this side, because on this side, that's where you have the ribosomes. The ribosomes are where proteins are produced. So all those subunits are produced on this side here, which is the inner side of the bacterium, on ribosomes. Those proteins produced on ribosomes will need to cross the inner membrane, and they do that through a transporter. So you have specialized transporters for those things. They're going to go through the inner membrane and through this transporter here, and at the exit of this transporter, they're going to need a chaperone, and that's this D protein here. Otherwise, they cannot fold, and they are immediately degraded. They need the chaperone to fold, and this means that in the space between the inner membrane and the outer membrane, you have myriads of those binary chaperone subunit complexes. So you recognize here the DA, DF, DK, DE, DG. Okay? So the chaperone is D. Now what happens next is that those binary chaperone subunit complexes are going to go to a site of assembly. You see this protein C here. It's a protein that is embedded in the outer membrane, and it's a nanomachine in the sense that it's a machine that will receive all the chaperone subunit complexes. That's where subunit is going to polymerize. That's where the pilus is going to be secreted. Okay. So the first one to go is first this one, okay, because this red subunit G has to be at the end, so it has to be assembled first. Okay. So this DG complex will go dock onto this outer membrane protein. By the way, this outer membrane nanomachine, we call it the usher. Okay, so this complex go dock onto this usher here, usher protein. That's the first one to do this, and it does that because it has the highest affinity of all the binary chaperone subunit complexes for the usher. It is also the only one that is able to activate this usher transporter, and I'll tell you about activation in a moment. Anyway, the next one is this complex here that comes in. F react with G here. The chaperone on G is released in the periplasm. Then you have K coming, so the DK complex come in. K react with E, sorry, I messed up already. The next one is E, because E is after F, okay, and you have several of these E subunits that are going to come in one by one until you have this DK complex, the green one, and this is what you're seeing here. So G has been assembled, F, E, several E's, and now you have K. Now that triggers the polymerization of A, that's signed subunit. So you have this DA complex here that will come in here. The first one will react with K, but all the other one will react with the A previously incorporated in the previous cycle of incorporation. So thousands of those DA complexes comes in, one by one, A react with A, and so on, until you have the DH complex here, H react with A, and everything stops. Okay? So at this stage, this is what we knew about the system. We tried to understand how it works, and the first thing we do was to solve the structure of this chaperone subunit complex, but what I'm going to tell you about that structure is valid for any of those that you're seeing here. So I hope most of you knows what a ribbon diagram is. It's a diagram, a way of representing proteins that goes through the main chain atoms of the protein. And so what you're seeing here in green is the chaperone, and you can distinguish two domains here, one here and one here. And the subunit is here. This is what I'm showing here in sign. Now, if you look at the surface, which is probably a more realistic representation of a protein, if you look at the surface, forget about the colors. I mean, if you had a microscope and look at this, you wouldn't see it in color. But forget about the colors. But what you see here in the structure of that subunit, so this is the pillar subunit structure, you can see this very deep groove here that crosses the entire structure. Okay? Now, having a groove is not so terribly special for a protein. However, the way this groove is produced is special. So what is shown here is what we call the topology diagram of a protein. What you do is that you take the protein, you split it apart, and where you see strands, you draw arrows, and when you see helices, you draw cylinders, and then you connect everything. And for us, structural biology tells us this is an Ig fold, immunoglobulin-like fold, Ig fold. However, that fold has a problem because in regular Ig fold, you would have additional sequences from the C-terminus of the protein that will form a strand G here. So in that gap, you would have a strand G between strand A2 and strand F. However, that sequence here is not there, and pillar subunits have a missing G strand. Okay, and as a result, you have this groove that's being formed here. You can see this groove here, and that is due to a lack of a strand. So now, as you can imagine, this is a very unstable structure. It's like walking on just one leg. It requires help. You can't do it readily. What the chaperone does is that it inserts itself into that gap. It donates one of its own secondary structure to complement the fold, the truncated fold of this pillar subunit structure. And if you want to look at the 3 dimensions, this is what you're seeing. Again, I'm going to try to not tremble too much or shake too much. You can see that strand from the chaperone here. And if you follow it, it's really in the groove of the subunit. So we call that donor-strand complementation. It's rather old work, but I had to explain it to you before I go on. Now, as you can imagine, this has tremendous implication in terms of pillars assembly because the chaperone is not part of the pillar. So chaperone has to go. You have this strand missing that has to be provided somehow. And the model that we proposed, which is roughly right, and I'll show you the details in a moment, is going to be shown in this animation. So what you're going to see here, you're going to see the chaperone dissociating. Okay, you can clearly see the gap here between this strand here and this strand. So that's due to the missing strand. So the chaperone is inserted into that gap for the moment. I draw your attention to this part of the structure here, which we call the N-terminal extension. It's just the N-terminus amino acid, 10 to 15 residues, which are at the N-terminus of each of most of the subunits. And you will see it plays an important role in the polymerization. And let me launch this animation. You can see the chaperone leaving here. You can see this clearly, that gap here. And this is crying for something else coming in. And what's coming in is the N-terminal extension, the N-terminus of subunit next in assembly. And this is what you're seeing here. You can see now it has inserted a bit of sequence, which is the N-terminal extension of this subunit in red. Now the chaperone has left here, so you will have to imagine another subunit inserting its N-terminate into that gap. So this process of strand exchange, we call that donor-strand exchange, because the exchange is between the strand provided by the chaperone with the N-terminal extension that forms a strand here to complement the fold. So you can imagine now, I think it might be clear that the pilus is a polymer of subunit in which each subunit, so one you're seeing here for instance, is complemented in trans by the N-terminal extension peptide here in red of the subunit next in assembly. Okay? So the process of donor-strand exchange, we went through a lot of different methods to explore it. Ideally, you would want to have a chaperone-subunit complex here, which you challenge with another chaperone-subunit complex. What I'm showing here is the N-terminal extension of this subunit in red here. As I've shown in animation, this thing here is going to fit into the groove of that subunit in blue. So it's going to make this interaction with the chaperone here leaving. We don't quite do this, although we have done it, but we have also shown that it's just as simple to do away with most of the chaperone-subunit complex structure, and only do this reaction with the peptide. Again, it's only 10 to 15 residue peptide here that is able by itself to attack this complex, dissociate the chaperone, and fit into the groove of the subunit in blue. And we study each of those species by mass spectrometry, something we did with Shinny Radford and Alison Ashcroft in Leeds. And what we've shown is that the donor-strand exchange reaction operates through a zip-in, zip-out mechanism, whereby the way this peptide attacks this complex here is by inserting one of its own residues, so one amino acid side chain, into a pocket at the entrance of the groove of this subunit here in blue to form a transient ternary complex that positions this peptide right in front of the groove and allows it to invade the groove, thereby destabilizing the chaperone strand, and the chaperone dissociates. So I will show you a movie recapitulating all this in a moment. But what I would like to show you now is what we know about this nanomachine here, the Usher here. So let me remind you what the Usher is. It's an outer membrane assembly platform. It captures chaperone subunit complexes. It allows the subunits to polymerize with each other. And also, it is a secretion pore through which the pilus is going to be secreted. And it's fairly large protein, 800 to 850 amino acid. It's made of several domains, as many proteins do. You have an N-terminal domain of about 125 residues, and we know this one is dangling in that space between the inner and outer membrane, and it's capturing chaperone subunit complexes. So that's where the chaperone subunit complexes are going to dock first. Okay? This domain bound to a chaperone subunit complex is shown here. The structure has been determined not by us, by a group at ETH. And here you can see this N-terminal domain here in ribbon representation. So this is what that domain here. You can see the chaperone in yellow, the subunit in green. That's the structure that provides the insight onto how the N-terminal domain captures chaperone subunit complexes. Our own work was to look at the translocation domain, the one that is embedded in the membrane and through which the pilus is secreted. And this was done by expression. You express the protein, it goes into the outer membrane, then you extract it, you purify the membrane, you extract the protein with detergents, and then you crystallize that. And the structure is shown here. So it's a very large, better stranded transporter. As you can see, 24 strands. It's the largest ever seen. And then, remarkably, it has a folded protein as a plug here. So it's a part of the sequence that you're seeing here between residue 220 and 325 is folded into a plug. And obviously the bacterium would want to have a plug there because you don't want everything that is outside to just stream in into the bacterium. You need a plug. But obviously this obstructs the passage of the pillars, so at some point this plug needs to go. Anyway, that doesn't tell you really how the usher works. To understand how the usher works, we produced and maybe this is a bit of a specialist jargon, really. But what we do very simply in many ways is that we produce the usher, okay, never mind what it's called, but we produce the usher, and a chaperone subunit complex, so 3 proteins together, hoping that in the membrane it will form something which is assembled and that is insightful in terms of function. So we did that, we expressed those 3 proteins, we purified them in some ways, I'm not going to explain that, in detergents, and we get a ternary complex. That means those 3 proteins are able to assemble. You have the usher here, here the subunit, here is the chaperone. And we were able to crystallize that protein here and obtain the structure. So the usher itself, this outer membrane transporter secretion nanomachine, is shown here. You have, just follow my cursor here. You recognize here the 24-stranded beta barrel, which is embedded in the membrane. You might recognize here the plug. Remember, this thing in magenta was inside. In the previous structures that I showed you, it's now outside. Now, the N-terminal domain of the usher here is here. And I told you that the N-terminal domain of the usher, we thought that was where the chaperone subunit complex should be bound, because we knew that was the primary recruiter of chaperone subunit complexes. However, we don't see this complex bound here. We don't see it bound here. We see it bound to these 2 domains here, which are the C-terminal domains. That's 2, CTD1 and CTD2, C-terminal domain 1, C-terminal domain 2. As you can see, the chaperone in yellow and the subunit in green are bound to those 2 domains, not this one. And obviously what you can see as well is that if I rotate, you can see that the subunit has inserted inside the barrel, which was at the time and still is an exceptional view of a transporter. So anyway, the bottom line here is that this structure, at least one of the implications of the structure is that we have identified that there is a second chaperone subunit binding site formed by the CTD1s and CTD2s by the 2 C-terminal domains. So we now have the knowledge of 2 binding site. One is here, and I showed you the structure before. We know that this domain can capture chaperone subunit complexes, but this one can also bind chaperone subunit complexes. So how do they work together? Well, we explored that by superimposing those 2 structures. I showed this one before. I showed you this is a structure of the N-terminal domain bound to a chaperone subunit complex. So we know how this NTD here captures chaperone subunit complexes. So we can just simply superimpose that structure to this one by simply using the N-terminal domain here and here. Okay, you just superimpose that blue thing to that blue thing here. And you just look at where this structure lands in that one. Okay? And this is what happens. So what you're seeing, I obviously changed the color. It's no longer that what you see in green here is now showed in orange. But that structure here of the chaperone in ribbon diagram and this plus this is exactly the same as what you're seeing here. Okay? And what's remarkable is that what we find is that there's no clashes between the orange subunit and the green subunit. And that means that in the position we see the N-terminal domain, it means it's poised, it's ready to capture the next chaperone subunit complex in assembly. And moreover, if you look into the zoom in here at the interface between the orange and green proteins, what you find is what you see here. So again, different representations. This is ribbon instead of surface, but it's exactly the same structure. And you can see that this N-terminal extension of this subunit in orange is right near the pocket here at the entrance of the groove of the subunit in green. That means everything is ready for reaction to occur. So in our model, the structure that you are seeing here is poised to receive the next chaperone subunit complex in assembly, and that's what is shown here. This positions those two subunits in orange and green for the donor strand exchange or strand invasion. This is what you're seeing here. The chaperone that was on the green subunit now dissociated. This site is free, so everything's going to move there. Here we are. Now this is free, the N-terminal domain is free, can capture the next chaperone subunit complex in assembly. Again, this positions everything perfectly for donor strand exchange and then transfer. We have experimental proof of this transfer reaction, so don't worry too much. I'm not stretching the things too much. So, and so on and so on. So I'm going to probably, I'm going to show you this movie here that recapitulates everything we know. So this is the structure that I just showed you. You can see the better barrel here. You can see the plug here, the N-terminal domain here, the CTD1, CTD2. You can see the chaperone subunit complex. And what you're going to see here is a chaperone subunit docking, reacting, transferring. The docking to the NTD, reacting. The white part is the N-terminal extension of each of the subunits. So you can see it fits here. Reaction, and then transfer. Now we're going to zoom in to look at the zip in, zip out phenomenon. You can see the P5 pocket here into which you have this residue inserting in that positions the N-terminal extension in white for zipping in through, and the chaperone leaves, dissociates. Now, why do you have a unidirectional translocation towards the outside? It's going to be shown now. You have indeed a quaternary structure, if you wish, of the pillars that draws the entire reaction towards the outside. 1,000 subunits later, you have massive pillars here that you can see. We're going to look at termination. So the termination subunit is in red here. So the termination subunit, and you'll see in this zoom in, the termination subunit here is able to react because you have this P5 pocket here. You can go through it. You can have a reaction. However, the termination subunit has no pocket here. That means that anything that's going to try to bind here will desperately attempt to react, but will not be able because it cannot insert. I think my time is up, so I'm going to stop here. I thought I'd show you many more things, but let me just acknowledge the people who've done the work in a few seconds. So collaborations with a number of people here, but the people in the lab who solve those beautiful structures are Gilles Fine, William Allen, and Sebastian Geibel. And I thank you for your attention.